Almost 3 out of 4 patients had stable disease after the first dose of SON 1010, and nearly half of the patients continue to have this stable disease for 4 months. We're encouraged by this type of evidence that SON 1010 is not only safe, but it can also lead to tumor shrinkage like the partial response we saw at the highest dose.
Welcome back, everyone, for another Virtual Investor What This Means segment. My name is Janine Thomas. I am CEO of JTCIR, and I will be the moderator. Today, we are very excited to welcome back Dr. Pankaj Mohan. He is Chief Executive Officer, and Dr. Richard Kenny. He is Chief Medical Officer of Sonnet BioTherapeutics. Welcome back, gentlemen.
Thank you, Janine. We are very excited to be here too.
Well, so happy to have you. And before we get started, I just want to inform our audience that Sonnet BioTherapeutics Holdings, Inc. is listed on NASDAQ market and trades under the ticker SONN. And during today's discussion, the company will be making forward-looking statements. And I encourage everyone to view the company's website at sonnetbio.com or the SEC's website for their latest filings and information. So, gentlemen, we are going to dive right in. Pankaj, first to you. You can explain what is the significance of Sonnet's new data release on SON 1010?
Thank you, Janine. Yes, this data release on our lead program is a significant milestone for Sonnet's clinical development. We have now successfully completed dose escalation in our first trial with SON 1010 with cancer patients, and are pleased to see a partial response in one patient at the highest dose level. In addition to clinical benefit in almost half of the patients evaluated. Safety of this extended PK version of interleukin 12 has been within expected levels, and the comparison of serum levels versus dosing in healthy volunteers, a study previously completed by us, provided strong evidence of tumor targeting in humans. We have used this trial to establish the maximum tolerable dose, or MTD, and will continue to follow the patients currently being treated to assess long-term safety and tumor responses. Sonnet is currently seeking partnership opportunities to help support later stage development of SON 1010.
Thanks for that, Pankaj. Rick, can you put into context the importance of the initial safety and tolerability data for investors, and what types of issues have derailed IL-12 programs in the past, and how has Sonnet developed SON 1010 to overcome these issues?
Sure. SON 1010 is actually a single chain variable antibody fragment, also called SCFV, that's been designed to target albumin in the serum. The drug can then be carried to cancer cells by using two receptors, GP60 and FcRn, the neonatal Fc receptor, that are overexpressed on a lot of solid tumors. And it stays in the tumor microenvironment, or the TME, by binding to a molecule called SPARC. Our SCFV candidates each contain a fully human albumin binding domain, or an FHAB, that binds to native albumin in the systemic circulation. Everybody has this. And this allows us to deliver SON 1010 subcutaneously and systemically, as opposed to injecting it directly into the tumor. Once this FHAB binds to albumin in your serum, it's preferentially taken up into the tumor microenvironment through these overexpressed receptors, GP60 and FcRn. That's what creates the targeting approach of our molecules. Inside the tumor, it binds to SPARC, and which prevents it from diffusing out of the TME, which allows for the prolonged half-life and the extended tumor retention of our molecules. We've designed our constructs so they preferentially bind at the lower pH that's also often found in the tumor microenvironment. We've created a targeting, delivery, and retention platform that was designed to deliver these payloads to the tumor with the goal of avoiding off-target activity, which might affect healthy cells in the rest of the body. With SON 1010, we're delivering the cytokine called IL-12, interleukin 12, into the tumor. As we've disclosed, our preclinical mouse data suggests that we target these cancerous cells with about a 35-fold greater potency than wild-type IL-12 in a mouse melanoma model. Interleukin 12 activates both T and NK cells to produce interferon gamma. It also converts the local M2 macrophages to M1 cells, and it enhances the anti-tumor effects, both in the innate and the adaptive immune systems. Previous efforts to deliver systemic IL-12 haven't really been successful because they're not targeted and they can cause off-target effects, such as cytokine release syndrome. Now, CRS is an immune reaction where the release of large amounts of cytokines can cause systemic inflammation and symptoms like fever, low blood pressure, and organ dysfunction. It can occur after certain immunotherapies and limits the dose that the patient can tolerate, or causes the patient to miss or skip doses until the CRS resolves so they can continue their chemotherapy. What we've seen so far in our phase 1 study is that we can dose SUN 1010 without causing any dose limiting toxicities or cytokine release syndrome.
Thank you for that, Rick. Now, the maximum tolerated dose identified was 1,200 nanograms per kilogram. Can you help investors understand how that dose in humans compared to your preclinical data?
So you can't do a direct comparison of the dose. But as we've shown in our preclinical models, SON 1010 has about a 35-fold increased potency compared to wild type IL-12 at reducing tumor growth in this mouse melanoma model. So we can deliver a lower dose of SON 1010 to enhance the safety with a hopefully stronger effect. With the new data from our SB101 trial, we now believe that we're delivering a therapeutic dose, and we're starting to see the translational data that supports this hypothesis.
Rick, this seems very encouraging. And this phase 1 study enrolled patients with advanced solid tumors and investigated SON 1010 as a monotherapy. So, approximately 50% of the patients achieved a clinical benefit, and you saw one patient with a confirmed partial response based on RECIST criteria. As you know, this is a What This Means segment. So, can you explain to investors what this means?
Our study SB101 is a very early stage, phase 1 dose escalation study, and we're enrolling late stage cancer patients, most of whom have already exhausted their prior approved therapies, and they're now seeking alternative therapies in clinical trials. Most of these patients have failed their multiple lines of prior therapy, including checkpoint inhibitors, and have progressive disease when they start our trial. When we see stable disease after 4 months, it's encouraging because we think it's indicative of SON 1010 activity in this trial. If you can take a cancer patient and stabilize the growth of their tumor for several months, it's a standard measure of clinical benefit. Almost 3 out of 4 patients had stable disease after the first dose of SON 1010, and nearly half of the patients continue to have this stable disease for 4 months. We're encouraged by this type of evidence that SON 1010 is not only safe, but it can also lead to tumor shrinkage, like the partial response we saw at the highest dose.
Well, Rick, encouraging indeed. Can you explain what the next steps for SON 1010 are?
Now that we think we've identified a safe dose of SON 1010 in cancer patients, we can start to expand the study to explore longer term dosing and identify what specific tumor types might best respond to our drug. We'll explore SON 1010 in earlier stage cancer patients with potentially healthier and more active immune systems, so there's potential to see better results in the future. In addition, interleukin 12 has been shown to increase or upregulate the expression of PD-L1 on solid tumors. So we believe this allows us to dose with a PD-1 or PD-L1 inhibitor and see a potential synergistic effect. We're currently exploring the combination of SON 1010 with a checkpoint inhibitor in advanced solid tumors and ovarian cancer in our study called SB221.
Well, gentlemen, certainly exciting news. Congratulations again. And with that, this concludes the Virtual Investor What This Means segment with Sonnet BioTherapeutics. And I'd like to thank Dr. Pankaj Mohan, CEO, and Dr. Richard Kenny, CMO of Sonnet, for joining us today. As a reminder, Sonnet BioTherapeutics trades on NASDAQ Capital Market under the ticker SONN. And if you like what you saw today, I encourage you to visit sonnetbio.com for more information on the company and to sign up to follow the company to receive their alerts, as well as follow their social channels to stay current on the latest information. And you can also visit virtualinvestorco.com for our latest segments and event calendar. I'd like to thank you for joining us and wish everyone a great rest of your day.